Yeah, so again, this is Xiaojie. I'm a postdoc from Jonathan Weissman's lab at the Whitehead Institute at the MIT. So first of all, I'd like to thank for this interview. I mean not the interview. I mean for this talk. And today I'm gonna tell you my story on Dynamo for predictive modeling of single-cell genomics. In the last few minutes, I will also tell you another story on Stereo-seq, an impressive spatial transcriptomics technology. And both papers has been recently accepted by Cell or published. So for many years, I have always been interested moving toward predictive modeling of biology. So I want to start with a very theoretical view to motivate how I think about biology. In the discovery of the fundamental principle of astronomy, Tycho made the first comprehensive observation of planetary movement, which leads to the derivation of the empirical laws by Kepler. And finally, to the unifying theory of gravitation by Newton. The key question is, can we also able to obtain theories of biology from big data? Specifically, similar to astronomy, who like to understand how planet moves, I always wondered, how does a single fertilized cell give rise to an entire organism with trillions of cells in a precisely spatial temporally controlled manner? So now groups in UW, groups in broad, and groups always world has generated many cell atlases that really provide the unprecedented view of the transition from progenitors to mature cells across the entire organism. So it's clear that biology is actually at this phase of searching for a unifying theory. And what I'm really excited about is that can we actually go beyond these cell atlases to predictive models of cell-fate transitions. This really began 10 years ago when I was a master student and I was fascinated by Conrad Worthington's epigenetic landscape, an elegant metaphor of developmental process. At that time, I made extensive theoretical efforts to quantify the epigenetic landscape. I started with writing down the ordinary differential equations of the canonical torque switch model, and then I mathematically mapped the epigenetic landscape and also quantified the associated development, dedifferentiation, and transdifferentiation paths. Since this work was all theoretical, at that time, I always wondered how can I quantitative model the actual genomics data that was just started to emerge 10 years ago. This was exactly what I delved into for the next 10 years or so for my PhD and postdoc. During my PhD and postdoc, I made a substantial contribution to the computational methods development in single-cell genomics. In my PhD, I wrote a monologue 2, monologue 3 to robustly and accurately infer the developmental trajectory from single-cell genomics data. Also wrote a script. It's a method to learn the causal regulatory network. For my postdoc and developed Dynamo that I will talk extensively today. On the other hand, my computational expertise also made critical contributions to break through technology development, including scale attack-seq or scale RNA-seq, and also the first droplet-based metabolic labeling enabled single-cell RNA-seq, and recently the Stereo-seq. So Dynamo really begins with a concept that attempts to derive the equations for cell-fate conversions. As we know, just because we know the a theory of gravitation, we can predict how the planet moves in the space over time. Similarly, if we can derive the equations of self-fed conversion, I believe we can predict how cell moves in the gene expression space. In my view, there are two major efforts in the past few years that moved towards this overarching goal. The first one is pseudo-time ordering with the machine learning algorithm Monaco 2, Monaco 3 that are developed in my PhD. We can leverage the gene expression similarity across cells to learn a graph manifold and all the cells along this manifold, so we can quantify the relative position and branch of each cell in a dynamic process. Another exciting development in the field is the introduction of RNA velocity in a very creative work. Mona, Zhang, and Peter and others show that by leveraging the incidentally captured introns from single-cell RNA-seq, they can derive a measure of RNA velocity, basically the time derivatives of the spliced RNA for each gene in each cell. They can then project this high-dimensional RNA velocity vector to low-dimensional space to reveal the local dynamics for each individual cell. So RNA velocity is really one of the landmark papers in learning the cell dynamics in the single-cell genomics. Nevertheless, both short-term, I wanted to say both short-term method and also RNA, let me go back, and RNA velocity are limited. Short-term methods only reveal the central trend of the gene expression kinetics, visualized by this thick principal graph in the middle, while RNA velocity, including just exciting developments, mostly will only tell you the local dynamics of each individual cell, visualized by the queries shown here. Almost about 4 years ago, when I was thinking hard on this, I wondered, can we actually go beyond just discrete RNA velocity vectors to construct a continuous equation of self-weight conversion? And importantly, it's obvious to me that once we have such an equation, it will open doors to many fundamentally different analyses that are not possible with RNA velocity and pseudo-time. For example, I want to use this equation to predict the cell fate for each cell. I want to use it to resolve the regulatory network and then also predict the optimal reprogramming path and TFs, transcriptomic factors. And then lastly, but not least, I also want to use it to perform in silico perturbations. To really address the question of deriving the equation of self-weight conversion, we need to ask a few questions. Firstly, what are the best data for learning such an equation, and then how to learn it? Secondly, if, as I just mentioned, once we have the equation, I need to figure out how to actually achieve this novel, non-trivial predictions I just mentioned. So in the following, with the intention to address these questions, I'm going to walk you through the theory of our approach, and then I'm going to walk you through the data and the experiments, and then I'm going to show you how we can learn the equation, followed by some examples that I hope will be illustrative of how powerful this predictive model can be. Finally, I'm going to close with some my current research and tell you my future directions. So the real transformative introduction, transformative innovation of Dynamo lies in the fact that it marries the scalability with from the machine learning with the interability of dynamic systems and the differential geometry. So with the machine learning approach, they can learn the equation of the transcriptomic vector field for thousands of cells, hundreds of thousands of cells in a matter of minutes. Well, with the dynamical systems, we can identify the attractors, attractor Bayesian, starting point separators. We can also leverage the equation we learned with the machine learning to perform high-order differentials using differential geometry. We can use it to calculate RNA Jacobian, RNA acceleration, and so on. Each of those terms has deep biological meanings. Again, I want to stress that this marriage is especially unique and powerful because it reconciles the black box but the complexity of the machine learning with the small scale, but because dynamic systems are generally only used for a very small network traditionally, but also they can provide the deep mechanisms. So they reconcile these two conflicted aspects, and in my view, this is really one of the first in biology. Here I'm going to first explain some basic concepts of a dynamical system with a cartoon. Under the view of the dynamical systems, each cell can be represented as a point in a high-dimensional state space where each dimension corresponding to a gene. The time derivative or is a velocity x dot is a function of the underlying regulatory network fx. For example, for this particular cell S2, we have a complicated network involving thousands of genes. Although complex, such a network may include characteristic motifs such as the mutual inhibition of self-activation network motif, like the P1 GATA1 motif that plays key roles in the hematopoiesis. Assuming a noiseless system, the regulatory network function fx will determine the trajectory of the cell fate xt. In this particular case, all the three cell state S1, S2, S3, they come from different places, but they all eventually converge to the same final steady state or attracted state A1. In the next few slides, I'm gonna use the toggle switch model to demonstrate how we can gain mechanistic and predictive insights with different differential geometry and dynamic systems approach. As you can see, this three cell state S1, S2, S3, they're coming from different places, but they all converge to the same attracted state A1. In fact, this system has three attractors in total, A1, A2, and A3, each associated with a corresponding attracted Bayesian B1, B2, and B3. Based on the RNA velocity flow, you can see all the states in each attracted Bayesian, they all converge to the same attractor. And then the names or planes, if it's in a high dimension space, that separate each attracted Bayesian and the separatrix, on which we have the saddle points, just an unstable steady state. And you can see around just saddle points, the RNA velocity flow has a high curvature, indicated they are extremely unstable. I believe such kind of analysis on single cell may provide a fundamental definition of what is a cell type. So you may define a cell type saying all the states are corresponding to a particular attractor Bayesian. Now I'm gonna highlight another important concept, RNA Jacobian, and show how they can reveal cell state dependent regulation. In plain language, Jacobian is just a fancy way of saying, in this context, how does the velocity of a gene changes when you change the expression of another or maybe the same gene? And the important thing I wanted to highlight is that we need to actually have an equation, the FS equation, to do the RNA Jacobian, because even you can measure the RNA velocity correctly, until you learn the function, you cannot take the derivative. In more details, let's focus on this particular dashed name. We can first plot the velocity of x1, f1 as a function of the gene expression x1, the gradient of this name corresponding to the Jacobian, and you can immediately see that the Jacobian has a positive peak, correctly revealing the self-activation from X1. Once we have this vector field function, we can actually calculate the Jacobian on any point in this space, not restricted to this dash name, and then we can use a heat map to visualize the Jacobian, and again we can see the cell state dependent activation from X1 to the cell. Also, I want to make a note that since we can calculate the Jacobian at any point in the space, so if we learn this equation, we can actually calculate the Jacobian for the places, for the regions where we don't even have the cell measured. Similarly, when we do the Jacobian from X1 to X2, again, we can also correctly reveal the repression from X1 to X2. Now I'm gonna also tell you that another interesting property is the divergence, which essentially is the trace of the Jacobian matrix. And if the divergence is positive, means the local flow it's like going out the set, so it's a source. When it's negative, it indicates in the sink. So, for example, in this particular data sets I'm showing here, we can see in the middle the divergence is positive, but on the side negative, because as you can see in the middle, just exactly corresponding to the progenitors, including like pigment progenitor others. While on the side, it corresponding to the terminal cell type, like Schwann cell and the iridoid and other cells. So next, I'm going to also show you that with another concept, which is the acceleration, which basically is a secondary derivative of the ion velocity, and then we can use acceleration to reveal interesting biological insights. So in this particular case, previously, when we use a conventional approach to analyze and annotate the cell states with the conventional marker gene detection approach, we are actually not able to identify the identity of this particular cell group. So we label it as unknown. However, when I try to use Dynamo to analyze the data to learn the vector field and also to calculate the acceleration, and can then take the top accelerated gene to perform Go enrichment analysis, and we can see under this analysis, I'm able to reveal just cells are potentially coming from control sets. Indeed, follow-up experiments confirmed just cells may indeed coming from the control sets. So now I mentioned a little bit the theory and also a little bit on the application of this theory. But what I really wanted to do now is to tell you what will be the best data for the measurements for has made fast to make for the purpose of learning the equation. So it turns out that the measurements seem to be the metabolic labeling enable the single-cell RNA-seq. There are many ways to do it, including one that get involved with the SCNT-seq with the Wu group in the Wu group from UPenn. A very nice thing about RNA metabolic labeling is that they will directly tag the next RNA in a controllable manner. So it can, in my view, it will provide avoid the intrinsic limitations from the biased capture of the incidentally captured introns and the unknown suppression rate. Therefore, in principle, metabolic labeling-based data can improve RNA velocity estimation and also reveal absolute RNA velocity. So now I'm gonna demonstrate how Dynamo will actually overcome intrinsic limitations of the conventional RNA velocity estimation. Specifically, I used the SC and T-seq to generate a data set of the human hematopoiesis. So we grow the human primary HSPC or hematopoietic stem progenitor cells in the maintenance media for to allow it to have spontaneous differentiation. And then at day 4 and day 7, we take the cells out to do the first labeling followed by SCNT-seq. We then develop the DNAST, a tool to process the metabolic labeling data to generate a new total spliced and unspliced RNA for each gene in each cell. We then perform the spliced RNA velocity with the state-of-the-art as dynamic model. Interestingly, the spliced RNA velocity reveal a overall pretty messy flow, and the problematic transition from mature cell type like megakaryocytes, erythroids, basophils moving backward to the progenitor. On the other hand, when we use the labeling data, Dynamo automatically reveals a smooth hierarchical transition from HSCs to each of the like GMP-like or MEP-like cells, which will then ramificates into a neutrophil, monocyte, basophil, erythroid, and megakaryocytes. This really nicely recapitulated the known biology of hematopoiesis, indicating the superiority of metabolic labeling-based approach over the splicing-based approach. Another thing I want to emphasize is that since we know the labeling time of the cell, we actually get the absolute instead of relative velocity with the labeling data. And talked about the ideal data, the labeling data for learning the equation of self-weight conversion. Next, I'm going to tell you how we can actually learn the equation. As I mentioned, we use the dynamic systems view to understand the cell state transition. Since we don't really know the details of this regulatory complex regulatory network, we would like to learn it directly from single cell data. Fundamentally, this is a machine learning problem where we would love to take the velocity data that include the current gene expression state X and some local dynamic information such as the velocity like dx over dt to learn the continuous and differentiable vector field function. In particular, we can either use the conventional, like the 10x based single-cell RNA-seq or the metabolic labeling based RNA-seq. I call it 10 result metabolic labeling based scRNA-seq or tscRNA-seq. The difference is that with the labeling data, you may get a better RNA velocity, and also since, as I mentioned, we know the labeling time, we can get absolute instead of relative velocity. So we can take this velocity data to train our model, then we get the continuous vector field function, and then once we have this continuous function, we can use it to predict the trajectory of each individual cell, and do the normal analysis I mentioned. So our method really works across different systems, as you can see on this data set of I think it's a neuronal activation from the SCNT paper. We can reveal the progression of the cell during the neuronal activity. And also we can use it to predict the cell cycle progression from the SCNT-seq study. And you may already see that our prediction can be either going forward in time, so you can predict the future, but you can also going backward in time, so you can predict the history of each cell. In more details, the vector field learning takes pairs of gene expression state X and the corresponding velocity V for many cells, and then we try to find an optimal function in the vector field functional space. So on this space, any point is a vector field function. And as you can see among the function GH2, the function H gave us the optimal fitting because it output the vector field that has the smallest difference to the input data. Mathematically, a vector field function is represented as a summation of a series of basis functions weighted by the corresponding coefficient vector. We use the reproducing kernel to represent the basis function, each associated with a landmark point. For example, x tilde 1, x tilde 2. When we sum them the basis function up, we get the resultant smooth vector field function that feeds the input data base. Since we have a closed form of the vector field function, we can actually analytically calculate the ion Jacobian acceleration and so on. And this analytical differential geometry is actually incredible because we can, for example, we can calculate the ion Jacobian between thousands of genes in thousands of cells in a matter of minutes. Now with the equation, let's dive into some fundamental novel analysis that the pseudo-time and ion velocity cannot achieve, and use such novel analysis to gain mechanistic and predictive insights of cell-fate transition. Over the past decade or so, people who study hematopoiesis found that, interestingly, the megakaryocyte lineage appear much earlier than any other lineage. However, we don't really know the molecular mechanism behind it. Here, I'm gonna leverage Dynamo's vector field approach to tackle this problem. First, reconstruct the vector field function, and then identify the fixed points that are corresponding to each cell type. We can then predict the time from the HSC to each of the terminal cell type. Consistent with the single cell transplantation experiment, we show that again the megakaryocytes appear much earlier compared to any other cell states. And importantly, since we have the labeling data, we have the absolute velocity, we can actually predict the real physical time for the HSC to become megakaryocytes, which is around 31 hours. And again, this is very well in name with the single cell transplantation, HSC transplantation experiment. So now, in order to dissect the mechanism that drive the early appearance of megakaryocytes, we show that we plotted the FLI1, the master regulator for the megakaryocyte lineage, and also the expression of KLF1 master regulator for erythroid lineage. And as you can see, the FLI1 already has high expression in the progenitor cell state. And then we perform the Jacobian analysis from FLI1 to KLF1, as shown here. And you can see across all the cells, the Jacobian is negative, indicated that FLI1 actually represses KLF1. Additional Jacobian also reveals that the FLI1 has a self-activation. So, so in conclusion, the FLI1 tends to have high expression in progenitor, whose expression is also maintained by the self-activation and also repression from FLI1 to KLF1. Thus, the HSC will be biased toward to megakaryocytes and make it appear much earlier than other lineages. Ever since the discovery of the cellular reprogramming, it has become a fundamental question in biology to predict the optimal path and the TF combinations that convert from one cell type to another. Previously, I used the principle of the least action path, an important concept in physics to predict the optimal cell-fate conversion for a simulated system. Now, since we are able to actually learn the equation of the cell-fate conversion from single cell data, I wonder whether we can also leverage the LLAP to predict the optimal paths on real data sets. In the context context, in the context of the hematopoietic system, we know that the key factors for all the development process, as well as the the reprogramming and also transdifferentiation processes. So what we would like to do is we would like to tag these known genes and the conversions and apply our LAP method to validate the accuracy, and then we can use it to actually predict other unknown conversions. So as I said, the LAP is a really fundamental concept, but to understand it, it's quite simple. Let's say we want to start from HSC to become the megakaryocyte. In theory, it can take infinity number of paths. For example, we may suppose to convert HSC to become erythroid and then to become megakaryocyte. But this kind of path will be very unlikely because some paths may have higher probability than others. So essentially, the LAP tries to find the optimal path that has the smallest action but highest transition probability to convert one cell state to another. And in this particular case, the first path shown here is the LAP. Now let's actually have a look at the LAP from HSC to each of the terminal cell type. Interestingly, the LAP are not the shortest path. Instead, it follows the curvature on vector field function, as you can see here. Although the LAP is shown on 2D, it can be operated in high-dimensional space. We can use it to reveal the full transcriptomic kinetics of each cell state transition. What I'm showing here is the HSC to basophil conversion. Importantly, we can also rank the contribution of each gene along this LAP path based on the accumulative changes along the path from which we can assign a priority score for each transcription factor. For example, here we calculate the priority score for all the transcription factors, and we find that GATA2, RNA-seq, those are known factors responsible for basophil lineage are identified. Here I'm gonna quickly show you that the priority score of the known transcription factors, the known transdifferentiation, this is a transdifferentiation experiment, are mostly above 0.8 with 1. It's a perfect prediction. And then the rock curve analysis for all the transitions, including development, reprogramming, and transdifferentiation, has a AUC around 0.85, indicating the high accuracy of this approach. Given the success on validating with the existing reports, here I'm going to show you that we can actually predict all the transitions from this matrix, including just regions that are not previously reported. So it will be an interesting future direction to validate the gene and the kinetics from the prediction in the yellow boxes. To further highlight the predictive power of dynamo, we then designed an in silico perturbation method. So we wanted to introduce a perturbation to the system, which can be directly we say we introduce some perturbation to the system, then we can translate the perturbation to the underlying gene regulatory changes, which the regulatory network can be encoded with the Jacobi matrix, which can be calculated at any point for any cell in the gene experience space. This perturbation will then propagate in this regulatory network to cause a lot of downstream changes, and then eventually you can calculate. You can also visualize the instant change cell-fate divergence after this perturbation. Since we can calculate Jacobian for each cell, we can also calculate this instant changes for any cell in the gene expression space. And then we can use it to actually predict the cell fate divergence after this perturbation. So you will not say we are converted from here to this direction. And here I'm gonna highlight one perturbation prediction on this famous P1 GALA1 network motif, because we know P1 GALA1 they self-activate and mutually inhibit each other. So it behaves like a seesaw model. In particular, so if we say let's suppress GALA1, then the counterbalance between P1 GALA1 will broken and SPA1 will win, and then cell will bias toward to GMP lineage. And as you can see from simulation, the cells indeed move away from erythroid and MAEP cells toward to the monocyte and the neutrophil lineage. Similarly, I think if we suppress SPA1, you will observe the GALA1 will win, and then cell will move toward MAEP like lineage. And as you can see from simulation, cell indeed move away from neutrophil monocyte to become erythroid megakaryocytes. And interestingly, if we suppress both genes, the counterbalance is more or less maintained, and cells are more or less trapped in the middle, like in the MAEP cell state, as you can see from here. So I think I need a bit over 10, so I'm probably gonna just skip my special transcriptomics part and just leave end here. So I hope I convince you that after 10 years, my dream of quantitative modeling on the genomics data is finally realized within this Dynamo paper. In particular, address the many fundamental questions and proposed in the very beginning, and showed that the RNA metabolic labeling is based SCRNA-seq is currently the ideal data for learning the equation. I also showed that we can use the kernel method to learn the equation, and importantly, we can use this equation to predict the future state and also reveal regulatory network. And lastly, through our LOEP and in silico perturbation method, we can make non-trivial predictions. So I'm gonna just skip that and go to the acknowledgement slide. So with that, I'd like to thank Jonathan for all the support for during the past few years, which made it possible for me to work on this very exciting project of Dynamo for my postdoc. I also want to, of course, thank Chen, George for Angie, and many others from the Weizmann lab for all the helps on performing experiments and the other stimulating discussions. So my great thanks to the call first also from Yang Zhang from Jianhua Xin's lab, who contributed enormously on the theoretical part of the work. A lot of equations are derived by Yang. So without his efforts, this paper is not going to be possible. I also want to thank my PhD advisor, Cole, for his wonderful support during my PhD career to develop Model 2 and Model 3 that I briefly mentioned. I also want to thank Jorge from Eric Lander and the Vijay Sankaran's lab, who worked with the Chen to generate the SCNT-seq experiment of human hematopoiesis, which again really made our paper accepted the sale. I also want to thank Xiaojie from Bell Hub, who did the initial single-cell senescence experiment. And of course, I want to thank my other collaborators, including Hall, Longqi, Jay, June, Suran, Emma, Ian, Lee, and many others. So yeah, lastly, I want to thank my funding resource, Jumia Clinic, and HHMI. So thanks for your participation. Let me know if you have any questions. Okay.
好的，谢谢肖杰。
Okay.
你现在可以听到我说话吗？
Yeah, you guys. But let me try to search input data RNA velocity 我可以用中文回答了，简单一点。对，是可以是 RNA， 但是这个 RNA 可以是 10x 或者是 dropseq。当然最理想的是用我们那个 labeling 的数据。然后第二个问题，什么叫讲话？我不懂什么叫讲话。对，我们这个是对我们这个想法就是说，你有这个函数，你就可以用这种方法去 predict。但我不知道你说的讲话是什么意思。因为我们这当然可能就是说，我们要求的是这个数据需要有能够把整个发育的过程都能够 capture， 中间不要有 gap。如果有 gap 的话，我们这个预测不一定准，当然也可以给你预测。但是我不太清楚你说的过于讲话是什么。对，在实验上可能确实有了，因为实验上你可能有受到外界的环境影响，比如说spatial 的信息，我这里没说，因为时间的原因，或者是 cell-cell communication 等等的 signal 外界的，对，我们这都没考虑。但是我觉得作为任何的理论模型呢，你应该想到的是简化，而不是把问题变复杂。至少是目前在我们这个模型，我们这个数据里面，它的效果是非常准的。我们不仅研究了 HSC， 也研究了个 pancreatic endogenesis， 预测也是很准的。对，当然如果将来有别的数据，你可以把它 incorporate， 这是我将来的 future direction。OK， 还有其他问题吗？
大家任何问题可以，肖杰，你可以先念一下问题，因为
分叉 OK， 对，都可以，因为你可以，比如说你人为的，我现在随便找一个就举个例子好了，你看，就是你看这个地方它有分叉嘛，你可以就是在这个分叉的区域，你可以把这些 cell 挑出来，然后去看一下里面哪些gene的 curvature， 因为分叉肯定是跟 curvature 有关系的。你可以把这个 curvature 给它把那些高的，因为 RNA curvature 你可以是每个 gene 都可以算出，在每个 cell 里面每个 gene 都可以算出一个值出来，因为你可以把这个 curvature 拿出来，然后去做怎么说？你可以去做什么 differential curvature analysis， 不是说 differential expression analysis， 你可以说在这两个 cell type 之间，这个分叉点附近的两个 cell type 之间，它们哪是gene的 curvature 发生了变化所以你就可以你可以把这些找出来。当然你也可以做同样的，也可以做 acceleration 你可以把那个 Jacobian 也可以找出来，你把那个区域是gene的特别是那些分叉一般对应着那个 mutual inhibition self activation， 你可以把那些区域，我们其实有一个函数专门就是说在这个分叉区给你找出。这个你如果仔细读我们的文章，在附录里面有一段描述了怎么去找那个什么 mutual inhibition 的 gene pair， 通过 RNA 的 Jacobian 是可以做的。OK， 还有别的问题吗？因为你 branch 嘛，你要从左边到右，你看这个 branch 有些 cell 往这边走，有些 cell 往那边走，是吧？它肯定是一个是往左，一个是往右之类的，所以它必须要有 curvature。或者你看我们那个simulate system，因为就看这个就更清楚了。就比如说，就这个 system 你看。你这个 system 假设 cell 在这里，这个是前提的，然后它这个前提它要变成，它要变成变成 A， 变成这个或者变成这个是吧？它肯定需要，它中间肯定要做一个决定，它是往左边走还是往右边走？那这个左边跟右边，它就肯定要，它这个决定就肯定会有 curvature。 对我想再强调一点，就是我们这个东西，非常的 powerful，不像什么 RNA velocity 就给你画个图，你看一下它的箭头往哪方向走，或者那什么后来要是什么 cell rank 什么 ACV 了，他们到只是基本上只是给你一个图什么的。我们这个是真的是有这个函数，你基本上知道了任何发育的任何信息，理论上，因为你知道这个 cell 的历史是什么，将来是什么，你也知道后面的机制，你可以，我最后也说了嘛，你可以做 perturbation， 你可以你可以 in silico 去敲掉一个 gene， 看它对这个 cell 的那个影响，所以是非常 powerful 的。OK， 还有问题吗？potential 也可以算，因为其实有一个我们 potential 有好多函数，有好几种，有一个就是如果仔细读我们的那个什么 10 minutes for 什么 introduction for dynamo 这个documentation 里面说了有那个因为我以前我 master 我就是做这些方面的研究的。我画的那篇文章，最开始画的这篇文章就是专门搞那个 potential， 就是10年前的这篇文章，就是专门就搞 potential， 所以我当时知道很多方法，包括那个最开始那个敖平老师，还有那个汪静，然后还有那个水晃，还有最开始有一篇叫 BMC Systems Biology， 一个比较 numeric 的方法。在我们的 package 里面都可以，但是我不觉得那些方法就这篇文章我说的这篇文章这个东西这个图其实就是一个 potential， 它就是算出来的 potential。对，有好多方法里面也是都 implement 的了。但是我是建议有一个就最近的一篇方法，我在我的文章里面稍微提到，你如果仔细去读，就是那个。有基于那个叫什么呢 Xiaojie decomposition 的那个 potential， 我觉得那个要比其他的方法要好，因为以前的那些 potential 完全是理论模型，它可能对这种小的网络work大行，但是在我们这种复杂的基于这个 weight field 是高维数据，我们这个数据是高维数据，是几千个 gene， 有几十万甚至上百万的细胞。它 work 的不一定很好，但是那个我刚才说的那个 Xiaojie decomposition， 它是基于 graph 的一个方法，它可以感觉是挺好的。回到技术进行我说了，我们理论上可以把整个网络都学出来嘛。那个 Jacobian 就是它其实是一个我不知道大家有没有理解啊？这个 Jacobian 它其实是一个几千个 gene 乘上几千个 gene 的一个矩阵，而且每个 cell 里面我都可以给你算出一个东西出来。我的每个 cell 里面我都可以算出这个网络出来，几千个 gene 乘几千个 gene 的这个网络，所以是一个3维的一个 tensor， 就是比如说几千个 gene 乘上几千个 gene， 再乘上几万甚至几十万的细胞，这样的一个大的矩阵。我跟你展示的只是几个例子，因为你真的要讲的例子就说明问题。但是对，你可以就是比如说你还是跟我说的，你可以把2个 group 的 cell 拿出来去做那个Jacobian 的 differential analysis。你看这2个不同的 cell state 之间 Jacobian 发生什么样变化，你就可以找出哪些 gene 发生了整体变化。Settle trace 是秀的太难。我们这个方法跟其他所有方法理论上是完全概念上不同的方法，因为别的方法都只是学一个 shield time， 对吧？它没有一个函数，我们现在是有一个函数告诉你后面会描述这后面的机制是什么，像按照 velocity shield time 包括 monocle 这个你说的这个 settle trace， 它都只是告诉你说只是一个值，最后给你的只是一个说这个cell 它是多少的一个值。我们现在是学了一个 equation， 就是后面就你就可以做这些 prediction。我刚才说的所有的这些分析， 就我的理解，没有任何其他工作可以做，是吧？你看我说的这些问题，因为你需要一个函数，你才可以去做，比如说 RNA-seq 啊，就这些，就我刚才说的这个，你才可以去 predict 每个 cell 的 cell state， 是吧？你才可以把这个刚才说的那个基因身上 cell 的那个组织给学出来，然后你把这个LAP 给学出来，你然后可以做出 population。对 label 的数据可能好一些，但是这个方法理论上10x 也可以做，但我卡维也是它是 relative， 就是你比如说我那个some make cases 那个 prediction 呢，你就不会知道它是31个小时，你就知道你可能还是能够看到 make cases 在出现再早，但是你不知道它大概花了多长时间。我们这个方法可以把这个31个小时给算出来。这是根本上的 conceptually 跟那些 ion velocity 不同的地方。OK， 就欢迎大家去用我这个 package， 大家可以去应该看到我这个 package 在什么地方，就这里，大家欢迎去用，然后可以给我看有什么想法或者有什么问题之类的。对，如果你们喜欢 package， 也可以去 start 我们的 package， 这样我们就可以更有动力去改进它。OK， 还有别的问题吗？
好的，谢谢大家，再次感谢肖杰，虽然他现在听不到。
如果没问题，我们就差不多可以结束了。
好的，肖杰，那我们今天报告就到这儿。
OK， 反正有问题的话，大家可以给我发 email， 对。
好的。
或者你读到我的文章，你们再仔细读了我的文章，有什么问题也可以直接给我发 email。
好的，谢谢肖杰，然后也谢谢大家。非常抱歉，我今天没有起到太大的作用。好，谢谢肖杰。